Let me start that now. Really appreciate you taking the time to talk with me. I'll admit now that my ignorance in your field is very, very high. I had to look up some words when I was looking at reading your bio, uh,
Yeah.
but I'll still try and understand what we're talking about.
Yeah. Okay, that's fine.
So, Dr. And I'm pronouncing it right, Dr. Steenbrugge.
Yeah, that's fine.
I read that you're a postdoctoral researcher,
Yeah.
laboratory of biochemistry. Can you tell me how you arrived there, what your career path?
Yeah, sure. I actually graduated as a biomedical scientist at a master degree in Leuven, Belgium here. I was strolling around to find a PhD position. I was really interested into oncology. Um, and I got to know some people working at the Faculty of Veterinary Medicine here in Ghent. And it was really it struck me that they were also looking into human oncology. So actually what they're doing here, and that's the interesting part, and what really interested me to do a PhD, is that they try to find the bridge between the human oncology and the veterinary oncology. So they actually not only work on mice as the normal gold standard model to check out new compounds or new treatments that eventually end up in the human clinic, but they also try to bring those compounds to the veterinary clinic. So sometimes, for example, let's keep it very broad, right? You have some childhood cancers, for example, like leukemia or lymphomas. Yeah, these are very difficult to, let's say, replicate in a mouse model, which is normally done by most scientists. So in that case, we try to turn towards more pets that normally have a high incidence of lymphomas, for example. And they are like a kind of a spontaneous patient, just like a human patient. And yeah, dog owners, they are not really reluctant to test new treatments onto those patients, because otherwise there are no real treatments available for pets. So in that case, it's like a very easy to go path to try to experiment with new compounds without harming the pet, of course. It's always in consideration about will it be toxic? So we already test the toxicity in a way on beforehand. But in looking at from that perspective, these dogs, it can also be cats, for example, they are like the ideal model that you can really translate to human clinic.
More ideal
So
than mice.
yeah, actually even so, yeah, because they have the same environmental exposure. They sometimes even eat the same food as we do. So we are really close to our pets, right? And that's what made it interesting. So you can actually make a difference not only for the humans but also for the animals. So it's like a romantic view maybe, but we call it the One Health perspective. It's actually a really well-known concept nowadays. And with the COVID, this actually even came into a bigger picture with COVID coming from animals. Keeping animals healthy will also keep humans healthy. That kind of concept. That's what we try to purchase here, or try to, yeah, let's say, contribute to. So coming back to the career path, I got the opportunity to do a PhD on oncology. First of all, with a human point of view, so a human endpoint. And I'm specifically focusing on breast cancer because that was the real, let's say, focus of this research group, biochemistry lab. Um, we we validate, let's say, a new kind of mouse model to study breast cancer in humans. It's a different kind of injection technique. We call it the intraductal injection technique. Some labs do it, but it's not easy to do. So it took me a while to learn it.
Why is it
Uh,
not easy to do?
so what's the difference with other the golden standard fat pad model, we call it? So what you do is actually you inject the tumor cells through the nipple of the mammary gland, and in that way the tumor cells will arrive inside the mammary duct. So in like the ducts that also harbor, let's say, milk when you give breastfeeding, right? So that's where a tumor cell or a tumor, human breast tumor, normally starts. It's the same in animals. That's also where it starts. And what other or most researchers do is inject the tumor cells just besides the ducts, inside the fat tissue. And that's actually already a more advanced stage of breast cancer. So what usually happens is that the tumor grows inside the ducts, and then it breaks through the ductal barrier inside the fat tissue, and from there on, it can spread to other organs. So it's a more advanced stage when you put it inside the fat tissue than inside the ducts. Most researchers just inject it inside the fat tissue because it's easy to do. And most of the time, they're also just interested in a growing tumor. That's it, not really those early stages. But we try to replicate the whole process from early to a late time point.
And if you can replicate the whole process, that gives you more power to say we can apply this to humans.
Yeah, exactly. Also, these early stage breast cancers are a bit like covered with this model, let's say. But then again, it's not easy to do. So most researchers don't turn to that kind of model. And it also has some drawbacks, right? You need to have a lot of mice to have a good, let's say, replicate pool. Sometimes some injections don't work out the way you wanted it, just because you had a bad day, for example, can happen. So towards an ethical point of view, it's not the most ideal because you need to have more mice. But then again, looking at from a human perspective, it's best to have the best replicate model that you can have for a human breast cancer.
And Dr. Steenbrugge, how often do these types of studies jump to human human applications?
Yeah, we always try to have some human perspective here. So we have a really great collaboration with our university hospital, but also outside our university, we have a lot of hospital connections, let's say. For example, in Brussels, we have a very good collaboration there with the clinic that's really specialized in breast cancer. So what they have there is human breast cancer samples. They also treat the patients there. So we get firsthand, most of the time, blood from those patients, where we can check new biomarkers that we found in our mouse models. Then again, it's not coming directly to the patient already, but it gives new perspectives. It gives new leads, where we then try to find new fundings to eventually come to clinical trials. So it's very difficult to push towards those clinical trials because you need to have a very strong foundation for that. It takes a lot of years.
Yeah,
so
I was going to ask, have you witnessed that process of the jump from mice, cats, and
yeah, we are witnessing it right now, more or less, with other kinds of or other kinds of treatments, not the chitin, but like, for example, we have now a kind of a trial, let's say, towards childhood cancers. So looking into lymphomas, where they try to treat now first the dogs as a kind of first model, spontaneous model, if you can call it like that. It's not really a model, but we call it a model. And that's the first lead towards going to the patients afterwards in, let's say, 2 to 3 years. So that will be in very short time going towards the first patient. Yeah, maybe going towards the chitin, right? So
yeah, so how did you first hear of chitin and get involved with this substance?
yeah, so it's actually we were not directly interested into chitin, but we were interested into the let's say proteins that bind it. So the proteins that bind it, these are the chitinase-like proteins. That's the first part of the family. And the other part is the chitinases. So it's the chitinase family, we call it. And you have, on the one hand, enzymatically active members of the family, and enzymatic or non-enzymatically active. So what's the difference is actually the possibility to cleave or to cut the chitin into smaller pieces. So from an evolutionary perspective, what's happened or what happens is actually that in an infectious disease, for example, you get infected with any kind of bacteria or a fungal disease. Yeah, these fungi have on their cell wall, so the outer wall of the fungi, these harbor chitin. So the chitin is recognized by our bodies through these enzymes and these non-enzymatically family members. So the chitinases will cut the chitin into smaller pieces, and then the chitinase-like proteins, they recognize these chitin particles, and then they start to stimulate kind of an immune response against these fungi. So you need to look at it like from an infectious point of view. That's the way we started also. We got to know these chitinase-like proteins in a kidney situation, so a chronic kidney or an acute kidney disease, first of all in dogs. So that's where we validated chitinase 3-like-1 as a really strong member when having an infectious disease that leads to kidney disease. And so we picked up this protein, and then we thought like, maybe we can put it in a broader perspective to other kinds of infectious diseases and maybe into cancer. So we looked into literature, and this was actually a field that was booming at the time. So people picked up the chitinase-like family as a kind of an inflammation-associated protein in different kinds of diseases, not only cancer, infectious diseases, you name it. You can see it from a really broad perspective.
And when did you look at the literature? Like, what's the timeline here? When did you?
Let's say mid my PhD.
Okay.
Through
So
that's already, I mean,
4 years ago.
Yeah, some 5 to 6 years ago.
Okay, and you saw that the field was looking at chitin.
Yeah, you had this one big group, like from the cancer perspective, you have one big group that was really focusing into chitinase 3-like-1. That's a main family member of the chitinase-like proteins. It was the Jackie Lyons group, and from Brown University in the US. And so they were the ones that were really going into that topic, specifically cancer related also. And they saw that it was really upregulated when mice got melanoma. And they also found that the source was mainly from macrophages inside the melanoma lung metastases. So these macrophages secreted the chitinase 3-like protein in a kind of response to the growing lung tumors or the lung metastasis from the melanoma. So this was really interesting to see that there was indeed a clear link towards cancer. So we started to look at that protein in our mouse models for breast cancer, and specifically triple negative breast cancer. We're mostly focusing on those really aggressive ones that's mostly found in the younger women. So what we found is also that the chitinase 3-like-1 protein was clearly enhancedly expressed when you gave or when the tumor was growing, and in an advanced stage you had a higher level than in an early stage. So then we were also looking into the let's say the source of this protein, and in our models it was not clearly to be macrophages, it was more like towards the neutrophils. So you have different kind of immune cells that can produce this protein. It's very ubiquitously expressed. So not only immune cells, but also like fat tissue can produce it, or even muscle tissue. But in a cancer context, or at least in our models, we found it to be highly expressed by the neutrophils. And neutrophils are really highly present in breast tumors in our models, but also in a normal human breast TNBC, so triple negative breast tumor. When you look at it, you also have a lot of neutrophils there. And even when you give chemo, you get an even enhanced infiltration of neutrophils as a response to the chemo. Okay, apart from that. So we also checked the protein in human tissue, and we found also a high expression in neutrophils there. So our model is more or less really comparable, but with the human situation, let's say. And that was the idea about, okay, how can we maybe, let's say, manipulate this protein in a way that we can like destroy the tumor. So what this protein normally does is it heals the tissue. Okay, so looking at it from an infectious point of view, the body starts to produce this chitinase-like protein in a response to the infection. So it wants to heal the body. But looking at or when taking that kind of perspective to a cancer context, it actually stimulates the tumor, because it tries to, in the healing process that it tries to stimulate, it actually stimulates the growth of the tumor. That's what it does. It stimulates, let's say, cellular growth. And in an infectious context, that's okay, because you try to heal the wound, right? Mm-hmm. But in a cancer context, you're actually stimulating the growth of the tumor even further. So we wanted to tackle that process.
And does this happen across all tumors?
Yeah, this is pretty much, let's say, a straightforward process that you can see in different kind of solid tumors, like colon tumors, for example, lung tumors. It's the same kind of process. These kinase-like proteins, they stimulate the growth of the tumor, even though they don't want to do that, right? It's an evolutionary process to heal the body. But in a cancer context, the tumor actually hijacks that process to stimulate its growth. And that's why cancer is such a complex disease. You're tackling evolutionary processes in a way that, yeah, this is how nature works, but in a way you try to prevent that. So that's very difficult, and that's why cancer remains such a huge burden to treat. So what we try to do is first of all look into the literature. How can we maybe, I don't know, block this protein from doing this healing process in a tumor context? And that's where we found the chitin. So there was one publication from 2012. It was from a group in, if I'm not mistaken, Miami, US. It's a paper from Stefania Libreros. She's now an associate professor, I think, in Yale. Never got to meet her, but I would really like to meet her one day. So she was the first to use chitin in a mouse model for triple-negative breast cancer. And she found that it could, in a way, block the chitinase 3-like-1 protein. So this is the main family member that most people are focusing, because it also is expressed in humans. So it's the same protein in humans and mice.
When you say use chitin, do you mean manipulate the chitin that's already there, or artificially inject chitin? Because
yeah, this is a thing about, I don't know how much you know about the chitin, but in a way, chitin itself, it's easy to isolate. You can find it in shrimp cells or from the exoskeleton from insects. So in a way, you can think about it like eating bugs. You get already chitin into your body, right? Mm-hmm. Or even eating shrimp shells, although we never eat the shell, but it's inside the shell. But it's more, it's not that easy to really target a protein with just a normal chitin. You need to have a specific size in order to block it. And this is where the Libreros paper was really focusing into the size. So they found that you need a chitin particle size of about 1 to 10 micrometers. So it's pretty small in order to have this anti-tumorigenic effect. So once they give these small chitin particles in a solution, this was just like a PBS solution. That's the normal physiological fluid that we use. And you inject the mice with that, and that's like we call it intraperitoneally. So that's inside the fat tissue of the abdomen. That's the normal way we inject it. In humans, you can even drink it or inject it through the vein. That's also fine. But in a mouse, it's easier to inject it into the abdomen for easy uptake. So when you do that and you give it like 1 milligram every 3 days, that was their treatment schedule, and you get a real clear growth reduction of the tumor. So that's what we first wanted to replicate. Can we replicate this kind of growth reduction into our models? And we actually found the same thing. So we saw that giving this 1 milligram, but specifically the 1 to 10 micrometer size, that's important. You get this same growth reduction, and also, yeah, clearly blocking the kinase 3 like 1 protein that's also inside the paper.
Congratulations on your paper too. I didn't say that yet, but that's
Yeah.
your work. It's inspiring. So
Yeah, this was actually main part of my postdoc. Yeah, it was a real interesting topic also for human clinicians, because now they have like an idea about, okay, if we maybe provide this simple natural product to our patients on beforehand as a kind of early treatment, we can maybe prevent tumors from going really big or even metastasizing, or even help our immunotherapy that's now really booming. So yeah, that's what we also aimed for, like combine it with immunotherapy, because that's now the becoming more or less the standard of care treatment for these triple negative breast cancers. But the main problem there is that a lot of these triple negative breast cancers, they're resistant to it. And when you have a metastatic triple negative breast cancer, this is more or less kind of a death sentence for these patients. You can just give chemo, and maybe in combination with the immunotherapy as a new kind of treatment. But then again, only 10 to 20% of the patients respond to it. So we need to get that percentage higher, because these are really young women. Most of the time, they only have a very young child. So devastating for those families. So giving like this simple natural product can already boost a bit the percentages. Or that's what at least we find from the mouse studies. So it really works in collaboration with immunotherapy. You get rid of the resistance towards the immunotherapy. That's what we found in the mouse. And you can also boost the immune system. So instead of healing the wounds, you actually boost the immune system to fight the cancer. That's what chitin does. Now, always take into mind that these mouse models are a real exaggerated situation, right? You never get to see this kind of immense immune cell burden and tumor growth into a human patient. Most of the times, these patients are already treated with other kinds of treatments on beforehand, before they get the immunotherapy and so on. So whether it will work the same way as we see in the mouse in humans, that's still a big question. But I'm pretty sure that it can work, at least in part, to stimulate the immune system with or without immunotherapy. And this is an interesting thing to know, because this chitin suspension, as we call it, has actually already been used way earlier than we think. We get to see the first reports in Japan. So the Japanese government actually stimulated universities there to do research on this chitin suspension, because they found that when people drank the suspension, you can even buy it in the pharmacy there, it's already available on the market for a long time. And they found that specifically cancer patients in an early stage, you get a way better response to therapy, and even a reduction of the growth of the tumor. And we're talking about early 90s here when they saw this. So this field is actually already a long time ongoing and evolving nowadays towards the chitinase-like proteins. Because at that time, they didn't even know what is the target that we are really pinpointing with this chitin. This was just drink the chitin, and you will get a better healing towards cancer, or even stomach aches. Let's say it was for all kinds of aches that you felt. And nowadays there are still companies that sell this chitin suspension in a way like for treating the older people in our society against arthritis or arthritis, sorry, like muscle aches. You get a better healthiness. Like it's a very romantic image to sell the product. In a way, it's correct, but they don't really know what they're selling because they don't have the molecular insight into that.
Yes, yeah, there's even a chitin society in Japan that I emailed,
Yeah,
try and
yeah.
talk with them about their
It's a
research.
huge thing there, right? Because
Yeah.
they eat a lot of fish too.
Hmm.
And that's probably where they get the chitin from. And yeah, so it's a really booming business there. I also find an Iceland company that sells it in a kind of pill form. But the main thing there, and this is important to realize, is that there's a huge difference between chitin and the more deacetylated form chitosan. So chitin, that's the normal normal saccharide that you find on the cell walls and in the exoskeleton of insects. This is pretty much insoluble, right? So when you tear it down into very small particles, you still get a kind of an insoluble flake inside your flask. And that's why most of these companies turn it to chitosan. So they do a deacetylation when you look at it from a chemical perspective. And in that way, it turns soluble. So you can get it soluble into a kind of drinkable version, right? But the main thing there, the problem there, when trying to treat cancer with this chitosan derivative, it doesn't work the same way as it does with chitin.
Why not?
This is still a pretty much debated thing. There are some reports that side by side compare the chitin with chitosan, and they say that yeah, the chitin does do an anti-tumorigenic effect, but the chitosan can even do the opposite. How it does that, no idea. Like, I also didn't look very much into detail onto that. I just knew like chitosan, that's not the way to go. Way too much reports that say it doesn't work. The chitin, we have a clear lead there with the Libriros paper. Let's just try it. It's easy to do. The only thing that was difficult is to get it into the very small particle sizes, because the chitin doesn't easily, let's say, break down. It doesn't easily break down, so you need to have a very strong sonicator, we call it. It's like a buzzing system where you break down those long chains of saccharides into smaller oligosaccharides, and then you filter the suspension through a very small filtering system that can only let through the particles of up to 10 micrometer, and then you the remaining suspension that you get is up to 1 to 10 micrometer size particles. So that's how we produce it. And we just buy our chitin from a commercial vendor. It's Merck. Merck is the commercial vendor, and they have like these chitin flakes coming from shrimp shells. It's very it's super easy to buy, and also not that expensive. We have like 5 grams for, I don't know, 50 euros. That may look like a lot of money, but then again, it's like a real let's say aseptic product. So it's been made into very aseptic conditions, so you can use it in a lab and so on. So bringing it to the patient, it's going to be pretty easy in my opinion, because it won't be that expensive, which again is a really big advantage. And in my opinion, it's also not that easy to produce. So or it's easy to produce, sorry. if you have this kind of system where you can tear the saccharides down to smaller particle size in a rapid pace, I think that's easy to do. I think companies already have that kind of stuff. And you get a filtration system that allows you to select the smaller particle sizes. You actually more or less have it. So, but then I checked clinical trials, right? Whether they already did some trials on the chitin. I couldn't find any. So up until now, I think nobody is really focusing on trials towards breast cancer or cancer with chitin, small chitin particle sizes. You can find some trials about chitin on, let's say, allergies, when with respiratory problems, you can have an adverse effect of the chitin. That's a different kind of, that's asthma. So that's a different point of view. Let's not go into detail about that. But from a cancer perspective, I cannot really find a trial for chitin. So that was also really striking to me because it's so easy actually and more or less straightforward to do. So what we're doing here now is we try to not directly go to patients, but we try to bring this treatment to the pets first. Because they're again, easy to make, not that costly, and that's a huge point in the veterinary clinics. That's the cost of the products. You don't get any reimbursement for veterinary medical products like you have in the human setting. So owners are more or less reluctant to do any treatment because of the costs. And if you have like a very cheap product that still works fine and is non-toxic, yeah, that can already help a lot of people, right?
Yeah.
And going then to the human clinic won't be a big leap. That will be just like not a walk in the park, but I mean, it can go pretty fast forward.
So the next step for you would be to try it with cats and dogs.
Yeah, exactly. Trying to get funding for that is not easy. That's another thing. So we're trying to find some ways to do it, but yeah, difficult. That's where we're more or less a bit stuck now. But that's budgetary part, not really science part, but it's also part of the job.
So Jonas, I'll share with you how I stumbled upon chitin because this may be interesting for you.
Yeah.
So I live in Paris and I met a woman who does research with chitin.
Okay.
And I think she has a chemistry background.
Okay.
And
that's what you mostly see. People working on chitin are real, like chemistry wise. Yeah.
Yes, but she, and I'm curious to know your opinion on this. I looked at she has a video and is working on her PhD. She's using chitin as a vehicle to transport into tumor drugs.
Yeah, this is a thing.
But that's not what you've done. What you've done is
no.
chitin in the tumor, and that prevents the tumor from growing. Am I understanding you correctly?
Yeah. So actually, this is an interesting thing because it's also inside our discussion of the paper, very small part. Chitin is mostly known among scientists as a kind of a carrier, a kind of a capsule where you can put in other pharmaceuticals or other kind of compounds. And most of it, most researchers don't think about what the chitin really does. It's just a capsule, a kind of vehicle to transport our product. Okay, great. But haven't you realized that maybe the effect that you see is not only the compound, but it can also be a part of the chitin effect? So this is most of the time just forgotten. I mean, people don't know what the chitin does. It's good to have it. It's biodegradable. It's good for transportation inside the body. And of course, it's non-toxic. That's the most important thing. So that's why it's a very interesting carrier. So we also put a small part on this in our article just to say like, hey, think about it. Whenever you use the chitin again, don't only think about the effects that your compounds may have, but also the chitin may do. It can also have anti-tumorigenic effects.
Yes, and interesting. I didn't know that in Japan it was already being sold as a supplement.
Yeah, I don't know how it is right now, but at least like in the 90s and so on, like 90s, early 90s, you could just buy it. I've never been to Japan myself, but I would really like to see it. Give
Yes.
me like a chitin suspension. How much does it cost?
Yeah, and when you say chitin suspension, what do you
Yeah,
mean? Like, is it a liquid with flakes in it? Is that
I
the
think so. I've never seen it, but I would say it's like that. So I think they just tear the chitin apart, put it into a flask like a suspension with flakes, probably. Like, um, how can I tell how it looks? It's like a grayish kind of yeah. Uh, it looks like a mold inside your bottle, but it's like a grayish kind of flake
Hmm.
that you can see. I mean, like it falls down to the bottom of your flask once you just let it stand for an hour. So it's not really soluble, right? That's what I mean with the chitin, and you don't have that with the chitosan. Then it just gets into a clear solution, but it doesn't have the same effect as the chitin has.
Yeah, and so in your opinion, that chitin is better than chitosan in that it needs to be insoluble to do its work. Am I?
Yeah, at least I don't know why. Yeah, right. So that I think that's still a big question mark. But reports and literature at least tell so that it's better to use chitin instead of the chitosan. On the other hand, I also find new articles, like very recent ones, that use a very deacetylated form of chitin, also chitosan called, right? But you have different kind of percentages of deacetylation. So chitosan can also be a very broad family of chitooligosaccharides. We call it oligosaccharides because you have different particle sizes. You cannot say it's one type of saccharide. Oh, it has different sizes, so that's why we call it the chito-oligosaccharides. And that's where they also checked it inside the, I don't know by a heart. Let me just check because I have it on my computer here. They also injected inside the tumor this chitosan, and they got a real anti-tumorigenic response. And it also has something to do with immune cells being stimulated. So in a way, I don't think it's a black and white story. Chitosan may work, but maybe in specific contexts and when giving it in a specific kind of manner, like injecting it inside the tumor maybe, and not drinking it or injecting it intravenously.
Yes. So Jonas, I also want to share with you the goal for this interview in our article. So
Yeah.
my colleague, she's in California now, so it's 2 a.m. That's why she couldn't join the
Okay, no problem.
she's done medical articles before, and she's the one with the connection at WebMD. Do you know
Okay.
WebMD?
Well, I've heard about it before, but of course, yeah, it's more kind of American website, right?
Yes, and it's become really the go-to for people with medical questions
Yeah,
online that want a reliable source.
yeah, yeah,
Um, and the working title for the piece, if it's accepted, is How Shrimp Shells Can Help Cure Cancer, right? Kind of a
yeah,
complete.
great. Yeah,
But the questions that I sent this in the email to you that the editor needs us to answer before they give us the green light is, what's the data showing what's going on with chitin? So you mentioned the interesting example of it being consumed in Japan. Is that just what you've come across, or have you seen any concrete data where the Japanese government or an organization has said, look, we've analyzed people who drank chitin or chitosan for 10 years, and those people got tumors that grew much slower than people who
yeah.
didn't
It's
drink
very
it?
difficult to find these data. You can find some, let's say, descriptions in review papers, but it's very difficult to find these data in like a raw form.
Mhm.
And I've checked multiple times, but very difficult to find them. I
Mhm.
can check for this piece, right? If I can definitely find something.
Mhm.
Um, but at least, yeah, it's probably also because it's so long ago. And some of these articles are not really, I mean, like internationally distributed. And it's more like in Asian, yeah, journal probably. I don't even know whether it's in English,
Mhm.
but I can check for you whether I can find this real hard data.
Yeah, if
Um,
you come across it or if it's convenient. Um,
yeah, yeah, no, sure, yeah.
because I'm also, you mentioned, and I'm going to watch this interview again. You mentioned the Stefani, who you want,
Stefania Libreros,
yeah,
yeah,
yep, and someone at Brown University.
yeah, that's
Um,
Jack Elias. You will find a lot of
nice.
stuff about the chitinase 3 like one. He's still working on that a lot. Um, and what they do is also not really work with chitin as a natural product, but they use antibodies against the protein. That's more a specific way of targeting the chitinase 3 like one protein. But this is a difference with the chitin. What we do with the chitin is actually we target the whole family and not only one specific protein. And this may also be a beneficial thing because we actually, in the paper, compared this antibody treatment for this specific protein with the chitin treatment. And the chitin treatment was even more efficacious. So you get a better growth reduction with the chitin than with the antibody treatment. So it's better to actually target the whole family. But it's difficult to tell how it works on each family member. That's a whole different story. We specifically focused on this chitinase 3 like one because that's the main, investigated protein. We know a lot about it in cancer already, mainly through the Elias group. And it's another way of targeting it.
I see. So there's still a lot of mystery about exactly what's going on.
Yeah, we even don't really know the function of the protein. So we're actually still totally in the mist. I mean, we find a lot of leads, let's say, and we find that it's an interesting therapeutic target, but the exact function is still pretty unclear, even after all these years. It's even strange that us humans still produce this protein, even though we don't have a chitin like in the skin or something. So it's like, in a way, we lost the chitin throughout evolution. We don't have this exoskeleton or something, but we kept the proteins that degraded, probably because of a, I don't know, protective mechanism to boost the immune system once you have an infection. But in the cancer context, this is not a good thing, right?
Um, and could you clarify a little bit more about it's not a good thing that we have chitin when there's cancer or tumor because it helps heal the.
Yeah, so the chitinase 3 like one, or the family of chitinases and chitinase like proteins, they have a kind of a healing function, you know. So once you have a wound and an infection going on, the body will respond to that in the way they want to clear the infection, and then they want to heal the wound. And these chitinases and chitinase like proteins, they help in this healing process. So looking at that from a cancer perspective, the healing process is actually stimulating cellular growth. That's what you do in a healing process.
But then what do you do externally that either prevents that or encourages another process from occurring?
So that's where the chitin can turn up in a cancer context. You can use the chitin to block these chitinase-like proteins.
I see.
That's how you. And what's normally going on in an infectious disease is that the chitin from the cell wall of the fungi or whatever bacteria is entering into your body, it gets degraded by the chitinases. And here we have like an already degraded version of the chitin that we inject, and that blocks the proteins.
I see. So
It's a difficult way of understanding, I know, but
yeah, no, I
think
think I got
about
it
it
now.
as an evolutionary process that's normally going on when having an infection, and it's a good thing that we have these chitinase-like proteins to boost our immune system and heal the wound. But think about that in a cancer context, healing the cancer, that actually stimulates the growth. So we need to reverse that process, which is evolutionary and not difficult to do,
Yeah, and you can
or
only
not
repeat
easy
the
to do.
process
Sorry.
using the same substance.
Yeah, exactly. Yeah.
Okay, you've definitely clearly answered question two of the editor, which is what is chitin's potential. Now you've
Yeah.
out of
It
there's a
clearly
ton of
has an
potential.
anti-cancer potential.
But the last part that he asked us is
Mhm.
how far off is chitin from being used to fight cancer? So you mentioned the
Yeah.
next step, dogs and cats, but if you, let's say, have all the pieces fall into place, you get funding, you do more studies. If you had to say when would human trials occur, when would they?
I think it can go pretty straightforward or even fast forward because first of all the chitin suspension is actually already been used in people. I mean just willingly because
Yes.
people just buy it in Japan already in the 90s. But then again, you need to have the specific particle size, right, to be anti-cancer efficacious. Um, so whenever being able to produce these kinds of small particles in a large volume, because that's what you will need to do to give it to humans, right? The amounts that we give in mice will be much higher in humans. We also don't know how many times you need to provide it to the people. Um, we think it's non-toxic, but then again, are we sure? Specifically with these small chitin particle sizes. So these are processes that, or phases, let's say, that we first need to go through in order to really go to the clinical trial. But I think it will be, yeah, let's say if we start today, you can maybe already do the trial within 3 to 5 years, I think.
Excellent.
That's just like a broad estimation. Um, but yeah, the main let's say reluctancy that most scientists have is that it's probably not or you will not be able to patent the product because it's a natural
Right,
product
right.
and there's already a lot of patents around regarding the chitin. So that's why most researchers are like, okay, man, I'm not gonna stick too much time into that because yeah, we will
I make money
probably
from.
yeah, we'll probably not get a lot of money out of it. And also the same with companies,
Very
you
interesting. So
don't get a lot of money out of natural product that's already been used like for a long time ago in Asia.
mhm.
So I think that's a main thing why you don't see a lot of clinical trials going on.
Huh,
Uh,
that's fascinating.
but I don't have like any solid data on that, right? It's just like an opinion.
Because
That's
in the
always a thing.
yeah, because in the I never knew this in the cancer research field. Um, yeah, is it do people express that, look, we're not going to put tons of time and energy into a path that we couldn't have a lucrative um, endpoint, right?
Yeah, of course, we always try to go towards or go towards advantages for the patients. That's our main goal, right? But then again, um, in the setting of the scientific field, also with all the institutions wanting more money and so on, you need to have like I mean like an end goal of any kind of patent where like a return of investment can be the case.
And do companies or funding ever put that pressure on you and say, look, we'll give you money, but you need to tell us what that you'll have a patent at some certain time?
We
Um,
don't get like pressure from companies. That's not the case, but we get requests from companies to test compounds because we have good models, we have expertise in the field, and that's when we like from a total. I mean, we're like totally not involved into the research of those compounds. So we're like outsiders looking at it from a clear with it from a clear view, like no prejudices or whatsoever. Um, yeah, then we try to test these compounds into our mouse models. We give our results to them and let them know whether it does or does not work and what maybe the molecular mechanism is behind the compound action. So that's what we sometimes do. And that also helps the patients forward because those compounds are already mostly into clinical trials and so on. And yeah, there's a clear patent perspective there because they developed the compound. And sometimes we're also involved as an inventor because we unraveled the mechanism of action of that compound. That can happen. Yeah.
I see. And but you're not lucky, but you're in a good place being part of Ghent University's Craig organization because
Yeah.
can you talk a little bit about how you work with people there and your research with chitin? Are there a lot of other people working with you? Are you yeah, what's the atmosphere there that allows you to do your research?
Yeah, Krieg it's a great collaborative, let's say, team of different research groups here in Ghent University. So we have people working in the hospital on specific cancer types or specific topics in their field, like immunotherapy, cell death. I mean, like the microenvironment and how to manipulate it, radiotherapy, you name it. You know, you have a lot of expertise there. We also have connections with other institutions, of course, but Krieg is becoming a pretty well-known name inside Belgium and also outside of Europe even. I mean, so it's a great atmosphere for collaboration. We have a lot of collaborating teams. And we also have like a coordinator of the whole Krieg. It has a board with a lot of members. We also have, I mean, connections with the industry who know Krieg. And whenever they have like a request for additional, I mean, like testing of compounds or something or ideas, they first come to the chairperson or people involved in the board, and then they send it out to different labs that may have, I mean, like the tools or the expertise to help those companies. We also sometimes have a spin-off from Krieg that even happens. You know, if we find new compounds or new treatment candidates, it can happen that we eventually develop or some spin-offs develop from Krieg.
Excellent. And is everyone who you've interacted with at Krieg on board with chitin, meaning they're aware of your research, they think you're going down the right path, or are there some people who go, look, you're wasting your time. There are other things to be.
Yeah, no, people know the products, and they were really interested in the research. Specifically, the clinicians really they were really interested because they were like, this is an interesting way to like treat our patients in a non-toxic way. Can be cheap, can be easy to make. Yeah, this may be a really good way to go forward, even in different kinds of cancers. So it doesn't necessarily need to be used in breast cancer alone. It can also be used like in endometrial cancer or ovarian cancer, which has still a huge burden towards treatment. So yeah, there was a lot of interest in the research. We presented it also in our yearly meetings. And we get a lot of input from other groups that want to go forward with it too.
And this was recent, right? You must have presented it 2 months.
Yeah. So I also went to the US with a collaborating group in the University of Colorado, where we specifically also focused on the chitinase 3-like 1 as the family member that is the most important one, let's say, or as far as we know, or the one that has mostly been done research on. And that's where we checked how this protein is also involved into the metastatic process of breast tumors. And we found that the chitin can block the protein. That's what we already knew. But blocking this protein also prevents the tumor from metastasizing into the lymph nodes, specifically the lymph nodes. And it's like the first process that the breast tumor is going through once it wants to go to distant organs. So it first spreads to the lymph nodes, and from there on, it can spread to different parts of the body. And you can actually prevent or reduce that process by giving a chitin or blocking that chitinase 3-like 1 protein. That's where I specifically focused on there in the US. And I also presented my results on chitin there at the University of Colorado. There were also some people from the University of Texas there, MD Anderson. And they were like, wow, man, this can be really interesting because you can tell people like in an easy way that eating bugs can help them in preventing getting breast cancer. In a way that you can look at it from that perspective, it's true. But it's not that easy, right? You have the small particle size, the necessity of having those 1 to 10 micrometer sizes of the chitin. And also, yeah, you still need to get it digested. If it's still like a complete bug, your body needs to digest it into those small parts, and it's never going to happen. So you need to produce those small particle sizes and digest that.
And what's the word you're saying? Complete buck? A complete
Yeah, like a complete bug. I mean, like an insect that you
Oh,
eat.
bug. Okay,
This is
okay.
like, yeah, this is like the thing, right? Like a lot of proteins inside the bugs. You can fry it and you can eat them. So in that way, yeah, chitin is like a booming business. I mean, a lot of people start to eat this kind of bug food. And it's also a lot inside the new pet foods. So probably the pet foods nowadays also contain a bit of chitin. Cannot prove that, but I might guess that it is the case.
Yes. So it can be preventative by
Yeah,
just consuming it, but then
yeah,
also when there is a cancer
yeah.
and you can treat it.
So they specifically told this with the perspective of treating women that get pregnancy-associated breast cancer. This is a specific kind of breast cancer that that group in the US is working on. So these are like patients that pretty much quickly after the breastfeeding, they develop a breast tumor because the breasts, when stopping the breastfeeding, it like involutes. So the mammary gland is becoming smaller because the milk doesn't need to flow through again. So you get like a tearing down of the breast tissue. That's a normal evolutionary process. But in
Open
that,
for
yeah,
cancer.
yeah, in that evolutionary process, you can also get cancer because it's a huge like inflammatory process going on, and sometimes things go wrong. I mean, you can also have like a more genetic predisposition for that kind of cancers. But when having that kind of cancers, you most of the time you're a young female, yeah, and you have a bad prognosis because those cancers, they are pretty aggressive. So that's where they say like maybe we can give like the people that are genetically predisposed already and have the breastfeeding, you maybe already give the chitin as a kind of a preventive measure to prevent tumors tumors developing into those involuting breasts. And nowadays they already give like anti-inflammatory agents like ibuprofen, I don't know, NSAIDs as we call them. This is what they sometimes already use to prevent the growth of these tumors. But again, NSAIDs, they're not good for the kidneys and so on. So they have a bit of a toxicity when being exposed to them for a longer period. So chitin may be a good alternative, a natural product. I mean, yeah, that's where they were really interested in.
Yes, natural, biodegradable,
Yeah,
non-toxic.
exactly. Non-toxic.
Excellent. Well, Jonas, those are all the questions that I have. And I'm going to keep researching to try and find even more concrete data because when you presented at Colorado, were there any other people there presenting about chitin, or were you the only one? Like in the field right now, is it yeah, what's sort of the state of the field
With the
and
chitin, you mean? Yeah, it's
yeah.
still like difficult to find a lot of new papers. Whenever finding stuff about chitin, it's always in a carrier way, you know, like
Yeah.
using it as a carrier, as you said too. And it always turns out to be focused on the compound inside the carrier and never about the chitin. Chitin is just, okay, we used chitin as a carrier because it's easy to use, it's biodegradable, and it transports our product pretty well. That's it. End of story. But having real in-depth studies on the chitin, difficult to find. I'm
Yeah.
also constantly on the lookout, but even before our study, the only concrete study on chitin that we found was the Libreros paper
Mhm.
from 2012. So that's already like, I mean, more than 10 years ago.
Yeah.
And before this paper, we also published a paper on um, the use of chitin also, but in a mastitis context. I can also send you that paper if you're interested.
Yeah, I'd love it.
So that's actually like a mammary gland infection. So you can also like reduce that infection in a way by giving chitin. Again, blocking the proteins. It's like an immunomodulation. You cannot really prevent the growth of the bacteria. That's not what you're doing, but you're reducing the immune stimulation so that it doesn't get too exaggerated.
See.
Different kind of contexts. But I think you were mostly focusing on cancer, right? That was your main
Yes.
topic. So yeah, that's why I didn't want to go into detail about that.
Yes. No, but
Uh,
it's all about what the editor is interested in us exploring.
yeah,
And if WebMD decides that we don't have enough information, I'm going to pitch to other American magazines like New Scientist, Smithsonian, like art publications interested in cutting edge
yeah,
medicine and
yeah.
science.
Great. Okay. Yeah.
And
Did you will you need to have more input from me? Do I need to check some I mean articles or how do we proceed here?
oh well, all it all depends on just what you're interested in. So with the
Okay.
interview, I'm going to share it with my colleague. We're going to write up rewrite our pitch to her editor at WebMD and see what he says. Him or another editor may say we need even more, in which case I may reach out to you again and say either can we have another conversation or can you
Okay.
guide me um, on where we can find certain information. But I'm optimistic. I really enjoyed this talk, and you opened my eyes to really what's going on in this field. So I'm optimistic that this is enough. Um,
Okay.
but I'll be
You
in.
never think about shrimps the same way, you know.
Yeah, yeah. No, that's
Whenever you will eat shrimp, you're gonna think about, okay, if I eat the shell, I maybe be able to prevent some cancer here.
yeah, and that's sort of the interesting element is that this combination of it being just this simple right shrimp shell versus
Yeah,
something that could change how we treat cancer.
yeah,
Um,
exactly.
and
Yeah.
yeah, so I'm optimistic that this will be enough for the editor, but I'm not the one with the connection. It's my colleague who's worked at WebMD before.
I think there's one additional US group, but I don't think it's on the context of cancer, who was also focusing on like shrimp shells and turning into chitin. I can look that up for you. So maybe that's another connection that you can put inside the. I will send you the details if I can find it back, but I don't know by heart what's the topic about with the use of chitin. It was not cancer as far as I know.
Mhm.
Um,
Yeah,
and
no,
there's also a press release and stuff like yeah,
yes, yeah. When I originally wanted to work on this with my colleague, I wanted to explore things that you talked about. It's anti-inflammatory concepts, like all the broad applications, but
yeah,
usually the editors they want you to narrow it down to bite size. So someone on WebMD
easy
who's
to read. Yeah,
easy to
yeah,
read can just go, oh, what's a new cancer thing?
yeah,
And how shrimp shells can help cure cancer. But that being said, if I reach out to a different news organization or journal and magazine in the US, they might say, oh, we want you to do more. Um,
yeah.
but
It's
one, but
great.
one, but one more question, Jonas. You said early on in the conversation that you'd like love to talk to Stefani at Yale, who did the 2012 piece.
Yeah,
Do
yeah.
you, even though you haven't talked with them before, do you have any?
We emailed a couple of times. Yeah,
Okay. Why
but
why
that's
did
already a long time ago.
and why did they stop if they published this good article on chitin in
I
2012?
think they're focusing on they went through with the kite in a while, you know, focusing on this chitinase 3 like one protein too. But now they're looking into different kind of topics, not specifically cancer related. She also left the group and went to another institute because of postdoc and so on.
Okay.
So yeah, I don't know. Also, the chitin for us. it's like a part of projects. So it will probably, I mean, like return someday, but we're also not really looking into that now very specifically with other projects. We're more looking into more targeted approaches now, like for example, antibody treatments. Because most of the time, it's like I said, you need to have a patentable product. It needs to be very specific. With chitin, it's a really broad blockade that you're doing, and then again, specifically for like I mean, granting agents, you know, like funding agencies. Um, they really want to have this very pinpointed approach. And that's where you need to use antibodies instead of broad blocking chitin natural products, you
I
know.
see.
But we'll always like use it as a kind of a positive control because it has a better anti-cancer effect than targeting one specific family member. The thing that we're focusing now on is actually again to reduce this inflammatory process in cancer and stimulate the immune system, and also having a synergy with immunotherapy, where not only blocking this chitinase 3 like one family member is the main member of the chitinases, but also combining it with other protein targets, also stimulating the immune system. So have like a triple targeting approach.
Yep.
The more members you're blocking, the better,
I
but
see.
always in a very specific manner.
Okay. No, thank you for that. Now, you've also expanded my knowledge just on cancer, how cancer is fought generally. So
Yeah, I always try to tell students, because cancer is such a complex disease, you can never say like, oh, there's a tumor growing and your body starts to react to it and that's it. You always need to think about it in a way like it's an evolutionary process. It's like an infection going the wrong way. Your body tries to tackle the infection, tackle the protein, or tackle the tumor, sorry, but the tumor uses evolutionary processes to hamper that effect.
yes.
And
Yeah.
that's why it's so difficult to treat it. You're targeting evolution. So
Mhm.
nature will always find different ways to, I mean, like find other paths to still stimulate the growth of a tumor.
Yes. Ah, yeah. No, it's a complicated, difficult disease. Well, Jonas,
Yeah.
I have to run because I got to work. I got to work. I teach at 1 o'clock.
Okay. No,
Again,
no problem.
I
Yeah.
really enjoyed this conversation. I'll be in touch about how this progresses, and thank you for your research and what you're doing.
Yeah, no, thank you for this interview. It was very nice talking to you.
Yes. No, I'll be in touch.
Okay, perfect.